Welcome to The Explainer. Let's just dive right into understanding the fascinating and honestly highly specific clinical pathway of a very particular type of brain tumor. Today, we're decoding IDH-mutant astrocytoma. You can really think of this as a molecular detective story. We're going to trace the exact microscopic fingerprints this disease leaves behind, starting from the initial subtle clues in the patient all the way down to the tiny DNA errors that drive the whole thing. It's a brilliant example of how modern medicine has shifted from just looking at a problem to literally reading its underlying source code.
38 years old. I mean, think about that for a second. That is the median age of presentation for this tumor. It primarily targets patients right in the prime of their lives. It's an unsettling fact, for sure, but it's absolutely vital for our understanding. This isn't a disease strictly of old age. The vast majority of these patients present in their 30s or 40s. When a seemingly young, vibrant person presents with a brain lesion, this specific suspect jumps right to the top of the medical radar. And what's really jarring here is how sudden these symptoms can be for someone so young. Sometimes the tumor announces itself violently and abruptly through an unexpected epileptic seizure, but other times it's a slow, quiet infiltration. Patients might experience pre-existing, really subtle speech or language difficulties. And because these tumors frequently take up residence near the frontal lobes, we often see unexplained changes in behavior or personality. Tragically, those changes might be present for months or even years before anyone even realizes there's a physical mass growing in the brain. So this brings us to the core mystery of our explainer today. How exactly does a seemingly healthy young adult suddenly develop such a complex, life-altering brain lesion? Like, what triggers this biological betrayal? To answer that, we've got to investigate the origins, decode the physical clues, and ultimately map the precise fingerprint of this unexpected intruder. Okay, Section 1, defining the suspect. What exactly is an IDH-mutant astrocytoma? So the World Health Organization defines this as a diffusely infiltrating glioma. That basically means its cells spread loosely, almost like roots, into the surrounding healthy brain tissue. But the real key to identifying our suspect lies in its specific molecular markers. We're looking for mutations in IDH, ATRX, TP53, and critically, the absolute absence of a 1p/19q codylation. If you don't have this exact genetic combination, you are dealing with a totally different disease. This is the exact profile we're hunting for. Moving on to Section 2, the crime scene. Let's look at symptoms and brain imaging. Once a tumor is suspected, the first step is obviously advanced brain imaging. And this reveals one of the most fascinating radiological clues in all of neuro-oncology. We're comparing two different types of MRI scans of the exact same brain here. On the T2-weighted MRI, the tumor mass appears homogeneously bright or hyperintense, indicating fluid. But then look at the exact same mass on a FLAIR MRI sequence. It suddenly appears relatively dark or hypointense, with all these mixed heterogeneous signals. It glows in one view and fades in the other. It's almost like a radiological Jekyll and Hyde. We actually call this visual anomaly the T2 FLAIR mismatch sign. It's a visual hallmark that is highly suggestive of World Health Organization Grade 2 and Grade 3 IDH-mutant astrocytomas. Honestly, if a radiologist spots this specific visual mismatch on a scan, it serves as a massive red flag. It points directly toward our suspect, offering a really powerful clue before a biopsy is even performed. Section 3, the molecular fingerprint. Time to zoom into the DNA. Now, imaging only tells us what the tumor looks like from the outside, right? To understand why it's there, we need to read the genetic barcode. An overwhelming majority, between 83 and 91% of these tumors, share one exact identical genetic error. It's the IDH1PR132H mutation. It is incredibly specific. That small remaining sliver you see accounts for a handful of other rare mutations in the IDH1 or IDH2 genes. But really, this single misspelled letter in the patient's genetic code is the spark that ignites the entire disease. Let's walk through how this single mutation sets off a deadly domino effect, completely reprogramming the cell's metabolic state. Step 1. That IDH mutation causes the cell to overproduce a rogue substance, an oncometabolite, called 2-hydroxyglutarate. Step 2. This massive flood of 2-hydroxyglutarate triggers profound epigenomic dysregulation, resulting in widespread DNA hypermethylation across the cell's genome. And finally, Step 3, because of all this hypermethylation, important cellular differentiation factors are just completely silenced. The cell essentially forgets what type of cell it's supposed to be, favoring this stem cell-like tumor-forming state that's prone to uncontrollable self-renewal. But here's the thing, that IDH mutation rarely works alone. Alongside it, the tumor relies on secondary genetic accomplices to ensure its survival. Mutations in the ATRX gene induce abnormal telomere maintenance, which leads to severe genomic instability. Meanwhile, mutations in the TP53 gene actually disable the cell's natural self-destruct mechanism, preventing normal cell death. So what you end up with is a mutated cell that is highly unstable, yet practically immortal, allowing it to survive and just keep multiplying within the brain. Section 4. Under the microscope. Let's check out the histopathological evidence.
Under the lens, the evidence of the crime is honestly glaring. These neoplastic cells look visibly, well, wrong. They feature enlarged, irregular, and hyperchromatic, which just means very dark staining nuclei. They don't stick together neatly at all. Instead, they form this loosely structured microcystic matrix as they diffusely infiltrate right into the healthy normal brain tissue. You might even see them ballooning into plump, swollen cells known as gemmocytes with abundant glass-like cytoplasm. It is literally a picture of cellular chaos.
Section 5. Tumor grading and prognosis. Assessing the threat level. So the biggest question doctors and patients face is this. What actually triggers a grade 4 diagnosis, the highest and most severe classification? You might logically assume that a tumor needs to look aggressively destructive under the microscope to be labeled as the absolute worst grade. But let's see why that intuition isn't always true when we're dealing with modern molecular medicine. If we look at this grading matrix, notice how cell division or mitotic activity jumps up significantly from grade 2 to grade 3. But look closely at the grade 4 criteria. To reach grade 4, the tumor needs to show significant visual tissue destruction, like microvascular proliferation or necrosis. Or it requires a very specific genetic feature, a CDKN2A or B deletion. And right here is the absolutely crucial takeaway. Molecular biology overrides visual histology. Let me say that again. If a tumor has a CDKN2A and/or CDKN2B homozygous deletion, it instantly escalates to a CNS WHO grade 4, completely regardless of its visual appearance under the microscope. The cells could look relatively calm, but that invisible genetic deletion is an automatic ticket to the most aggressive grade, simply because we know exactly how viciously that specific genetic profile is going to behave. These exact classifications aren't just academic, they're vital, because the tumor's grade directly correlates with a patient's expected median overall survival. We're looking at potentially over 10 years for a grade 2 diagnosis. That drops to 5 to 10 years for grade 3. And if it hits grade 4, whether through visual necrosis or that invisible genetic deletion we just talked about, the median survival abruptly shortens to roughly 3 years. Getting the grade right means giving the patient an accurate, honest understanding of what lies ahead. This whole explainer really highlights how molecular markers have completely revolutionized neuro-oncology. We've moved from merely looking at the shapes of cells to reading the very source code of the tumor itself to predict its behavior. But it leaves us with a pretty profound thought. As we continue to map the molecular fingerprints of the brain, what other hidden genetic barcodes are just waiting to be found? And more importantly, how will discovering them unlock the next generation of targeted treatments?